We're really happy to have Ryan here with us today. And Janet and I met Ryan last spring when we took a bunch of students down to UW for a symposium and to learn about getting into the neuroscience graduate school. And Ryan and I just got talking about, what's it like to teach at a place like Western, and I kind of said, why don't you come up and find out? And while you're up here, you can give a talk. And so here he is. Ryan is a graduate student at UW in Paul Phillips' lab. He's just starting his 4th year this year, and he's going to talk to us about dopamine and neuropeptide. So, thank you.
Thank you.
Yeah.
Again, thank you all for having me here. Thanks for the invitation and showing me around the facility. It's awesome. I'm really interested in working at an institution like this further down my career, so it's great to see how it works here, and also all of the animal work and other research that's going on outside of animal models. So today, as he just mentioned, is that I'll be talking about dopamine, and I guess he didn't mention this, but dopamine and neuropeptides and how they regulate drug consumption. And just to start off, although most of you probably are going to realize that I don't need to have this spoken about why it's important to study drug addiction, it's always important to just recognize what's happening in our world and in our culture. And currently right now, or in 2016, it was estimated that about $700 billion a year were being spent just on drug addiction cases alone in the United States. And if you look at this figure over here on the right, there's an increase in the amount of overdose deaths that are occurring in the United States, and it's substantially increasing. And in 2016, it was about 64,000 drug overdose deaths we estimated. So this is not a problem that is not known. It's not a secret. We all know that there is a problem. It's especially prevalent with opioids. But two things that I want to focus on, because I look at animal models, is with animal models you can only look at certain or specific aspects of drug addiction. So the things I'm going to be focusing on today is one, compulsive drug taking, as well as loss of control and drug consumption. And I know some of you, if you were here, you've heard Jeff Grimm talk about some things about incubation of craving. And that's about how relapse takes place. I'm talking about the other side of things, where what about when the animal's actually taking the drug? And so just to get into the area or the brain substrates or brain locations that I'm focused on, I mainly focus on this nucleus accumbens right here. And why I focus on this area is this is like it's an integrator of information from other neural substrates that are associated with behaviors that are controlling actions with drug addiction. So such as the prefrontal cortex, which is known to be involved with decision making, the hippocampus, which is known to be involved with memory, as well as the amygdala, which is regulates mood disorders or mood regulation, as well as stress reactivity. And all of these things, all of these areas actually integrate and have glutamatergic inputs, which is shown here in red, into the nucleus accumbens core. So the nucleus accumbens itself, which is also known as the ventral striatum. So this region is set up in a location that receives inputs from all these different regions of the brain that are probably mediating and have been shown to mediate things that are associated with drug addiction. The things I'm going to be focusing on here is the ventral tegmental dopaminergic projections into the nucleus accumbens core, which is that green line here. So the VTA, which stands for ventral tegmental area, I'm going to use that acronym throughout the rest of my talk, as well as the NAC, which stands for the nucleus accumbens. And so what I mean is, what these dopamine projections really do, and why I'm interested in them, is that dopamine itself is a neuromodulator. And so what that means is that dopamine actually, when it synapses onto the main neurons within the nucleus accumbens, which are called medium spiny neurons, it actually synapses onto the dendritic spine, rather than this dendrite head here, where these glutamatergic inputs from PFC, hippocampus, and amygdala would come. And so it's set up perfectly to actually modulate or regulate what the glutamatergic inputs have and what that GABAergic medium spiny neuron outputs. And so the idea here is that dopamine is a modulator itself. All that means is that it can actually affect what the inputs into the nucleus accumbens are. So I'm really interested in what are these signals doing and how are they contributing to actions that are associated with drug addiction. And just to make note, because this is a mouse brain that I'm showing you here, the nucleus accumbens and the ventral tegmental area do exist in the human brain. It is phylogenetically preserved. So this is not just, hey, what does it look like when a mouse takes drugs? We actually care, what about when a human takes drugs? So that's why we're interested in these regions, is there is also imaging studies done in humans that show that this area is also involved in drug addiction in humans as well. And so just to go into why dopamine, is that dopamine actually, through the use of animal studies, this is using microdialysis here, and that line right there is actually when an animal received an infusion of cocaine. And microdialysis basically is a probe that can be put into the brain, and it allows the collection of how much dopamine was there at the time on a minute time scale. And so you can look at this, and it just does that by collecting the solution, and through passive diffusion can actually take up dopamine. You then can measure how much dopamine is there after the fact. And from this, and when they receive cocaine, you see dopamine goes up. And so this is the drug itself. And then over here is using fast scan cyclic voltammetry. It's a large word. I'll explain what that means in a second. But all this is actually looking at dopamine signals on sub-second timescales, so basically on 1 second resolution. So how fast does dopamine increase? And in animals that have actually experienced cocaine, this green bar here is a cue, or an environmental stimuli that's associated with the drug itself. And when that green bar is shown to animals that have experienced cocaine with that cue, they have this large spike in dopamine, or release of dopamine in this nucleus accumbens region. Whereas those animals that are naive or have not experienced cocaine to that cue have no dopamine released to that cue. So this is just showing that drugs of abuse, or particularly cocaine is what I'm interested in right here, and the cues that are paired with them are able to drive dopamine release within the nucleus accumbens. And so just a quick summary of why I'm actually studying this is that and the nucleus accumbens is that the nucleus accumbens really is set up in this area that can control all these different behavioral, or integrate all these different areas and signals in these areas that mediate these behaviors associated with drug addiction. And it's also preserved across all mammals. And animal models have shown that there's increases in dopamine to the drugs of abuse, as well as the cues that are associated with them. And so what do I mean by animal models? I've said that probably 10 times, and you might be confused. So just to go through what I mean by animal models is that we can take animals and we can train them to self-administer or pick when they're going to take a drug. What I mean by that is you can see on this rat here, this is a male Wistar rat. There's an IV catheter, so we implanted a catheter in its jugular vein, and that allows us to then deliver cocaine or other drugs of abuse directly into its veins, and then which will then be dispersed throughout the rest of the body. And so what we do is we have these animals where instead of sometimes they can press levers, we have nose pokes where the animal actually just sticks its nose in one of these little ports right here. One of them will be active, one's inactive, and when the nose poke into the active port, it actually leads to a drug infusion and a change in the environment, which what I'm calling cues right now, which is an autovisual stimulus in which a light comes on and a tone plays. So now the animal is I have it listed here as a CS because that noise, that autovisual stimulus, is becoming a conditioned stimuli. So now these animals are able to relate this tone and this light to the drug itself. And so what we can do with these things is that back in 1998, there was this paper that came out that showed, well, what if we give these animals more access to drugs? So this typically was done in 1 hour sessions a day. So you come into lab, you put the animal in for the animal in a box, they take cocaine for 1 hour, and then you leave. And this is what their intake would look like over time. However, if you give them which is here in these open circles, long access, which is 6 hours a day, over time they actually increase the amount of drug they're taking. And this is thought to be an animal model of the loss of consumption, which I mentioned earlier, is that these animals are actually increasing the amount of drug they're taking, and it seems like they're becoming more compulsive in their drug taking. And so the idea is we can use that and we can start studying what are the neural substrates and what are the neurochemical signals that are actually associated with those increases in drug taking over time. And so one way to do that is, I mentioned I study dopamine, so how do I study dopamine? Just to make a complicated thing that it's taken me 4 years of my PhD to somewhat understand and explain to you, is that we use this technique called fast scan cyclic voltammetry. And what that means is dopamine is it's a chemical that's electrically active, meaning you actually can cause it to go through a redox reaction, so you can oxidize it and then reduce it. And you may have or may not have learned yet, but when you actually go through a redox reaction, electrons are released. And so when electrons are released in a redox reaction, you actually can detect that electron release through current. And so what we do is voltammetry is we create an environment where dopamine actually gets oxidized, which turns into dopamine oquinone, releases 2 electrons, and then we can make the environment go back to what it was before by changing the voltage or the potential of that region of our electrode that's in the brain, and then have it pick those 2 electrons back up. And so what that really means is we're just allowed to use this fancy electrochemical detection technique to record the amount of dopamine molecules that are present. And so whenever I say, and what this outputs us with is we get this signal of dopamine. And what this is actually that yellow box here is corresponding to the cue with the drug infusion or the CS. So the animal nose pokes right here at 0 seconds, that cue comes on, and then we can detect this increase of dopamine. And so that's really all this technique is allowing us to do, is allowing us to detect dopamine at these sub-second time temporal resolution. And if you have questions about voltammetry, I'd love to answer them after, but just to keep going with what I'm interested in using this technique and what we can do utilizing this technique and the things we can measure. And so I just wanted to start off when I get into my data is to really, rather than just start showing you data without any idea of what I'm actually talking about, is to try to get you honed in on my thought process and exactly why I'm doing the experiments I'm doing. And so right here, this is my hypothesis that I've been working on for my dissertation, and I'm going to continue working on, hopefully for only 2 more years. And, so really what it is this is like a linear model, or it's a very simplistic way to think about this, but I think there's these neuropeptides that exist in the brain, and all that means is the neurotransmitter that's big. It's built out of a lot of amino acids that are connected by peptide bonds. And so there's one of them which is CRF, dynorphin, and then I'm looking at dopamine and looking at how that regulates drug consumption. So the whole hypothesis that I'm looking at is that I believe all these things are working in a linear fashion to then manipulate the amount of drugs that an animal consumes. And so this is a little bit better of a depiction of what I think it looks like in the brain, or at least in the nucleus accumbens, is that I think the CRF molecule acts on receptors within these neurons that exist in the nucleus accumbens, which then increase the amount of dynorphin, this other neuropeptide molecule, and which acts on the dopamine terminals and eventually leads to a decrease in dopamine. And I'm going to talk about why I think these things as I go through each one of the steps that I'm going to study this hypothesis. Okay, but just so you know what I'm talking about, I'm going to continue coming back to this model. And this might not make a lot of sense, but to start, I'm actually going to work in reverse order. So first I want to see how does dopamine, how are dopamine and consumption of drugs actually mediating each other? Does dopamine even mediate or promote an escalation of drug consumption? And so that's what I'm studying first. And so to do this, a postdoc in Paul's lab before I actually got there had shown that if you give animals access, he replicated this, where you give animals access to cocaine for 6 hours a day, they increase their intake over weeks. What he also found is that as those animals are increasing their intake over weeks, that dopamine signal to that cue that's coming on when the animal nose pokes is actually decreasing. So he's finding this correlation that, oh wow, look, when you look at how much drug the animal's taking, their actual dopamine signal to the cue that's associated with the drug is decreasing. However, he also noticed that only half of the animals escalated. So then he wanted to say, all right, so there's a separation in animals' individual drug-taking patterns. And so he looked at animals that do not escalate their intake versus those animals that do escalate their intake. And what he found is that the animals that are actually not increasing how much cocaine they're taking over weeks never had this attenuation of dopamine. Whereas those animals that did escalate did have an attenuation of dopamine. So this is just a correlation showing, all right, this is interesting that the amount of dopamine that's present to this cue and the amount of drugs or the amount of cocaine the animal is taking are correlated. So then what he wanted to do is, well, what if I can replace this signal in those animals that escalated their intake? And so what he did is he gave these animals L-DOPA, which is a precursor to dopamine, meaning that L-DOPA is required for dopamine to be synthesized. And so if you, this is a common drug that's used for Parkinson's patients because the Parkinson's patients have a hypodopaminergic state, meaning they have low amounts of dopamine. So what if you give them L-DOPA? Presumably you increase dopamine. So his thought was, well, what if I give them L-DOPA and I can increase dopamine? And over here on the right, you can see this is the nose pokes that is occurring in the second week, and the escalated animals are here in purple, whereas the non-escalated animals are here in orange. And you can see that L-DOPA actually decreases the amount of drug that the escalated animals are taking. that's great. So what he's finding is now he has this causal effect. So he has, all right, if I manipulate dopamine by administering these animals with L-DOPA, I see that these animals decrease their amount of drug that they're intaking. Whereas the non-escalated animals, because they don't have this attenuation, had no effect on their drug. Okay, so one thing that I want to get at though, and what I'm interested in, so I just showed you a lot of data from a postdoc, and now I'm going to start talking about data I've collected, is that what does L-DOPA actually do is that it increases dopamine total. It doesn't actually increase dopamine to this fine red temporal, this time scale that we're looking at. So although we can increase dopamine, we can't increase dopamine at the exact time that this cue comes online. And so when I joined the lab, I thought, well, what if we increase dopamine what if I just replace this signal rather than increasing total amounts of dopamine? And so that's the question I'm asking is, are temporally specific increases in dopamine release sufficient to reverse escalated cocaine consumption? And so the way that you can do that is through this fancy technique called optogenetics. And so what optogenetics is, you've probably heard a lot about this, or maybe you haven't, but I really think it's a cool story of how optogenetics came to be, is that I think in 1993 it was some scientists discovered that green algae has this receptor that responds to light, and it allows sodium to come in to the green algae or into these cells. And then some people had this great intuition is, what if we extract that protein, that receptor, and then we put it in other things? And that to me is like one of the biggest leaps I've ever heard, but has revolutionized the way we do neuroscience, which is really cool, is that you think, oh, I'm not really interested in green algae, but maybe you are. Because what that can do is that sodium comes in, and so sodium can come in, so if you put these in neurons and you shine blue light on it, which this sodium receptor is activated by, is that it lets sodium in, and then there's an action potential. And so that's what I'm showing here, is that every single time they shine blue light, there's an action potential that corresponds. So ideally, is what you could do with this now is if you get this in certain neurons, you can actually then cause neurotransmitter release by just shining blue light on these neurons. And so what I was able to do is I took that, other people have extracted this, and then I took all of that genius and the giants that came up with this technique, and I can stand on their shoulders, and now I can implant that channel, or that receptor, into dopamine neurons. And the way I did this is I used a virus, and so I packaged all the components, all of the DNA that's needed to have the cell machinery create this receptor, and then put them into the membrane. And so I injected that virus into those dopaminergic cell bodies in the VTA, and then I implanted a little fiber optic probe above the nucleus accumbens, so I could shoot blue light at it, shine blue light at it, and then I could cause action potentials, and presumably the release of dopamine. That's the idea, but can I do it? So this is showing some immunohistochemistry, and so what immunohistochemistry is I'm just using antibodies that have fluorophores attached to them, meaning that I use antibodies, they have things that fluoresce that can bind to them, and then I can look at how these neurons that are lit up or they're fluorescing. And so TH is tyrosine hydroxylase, which is another precursor to dopamine. So we assume that things that have tyrosine hydroxylase in them are able to synthesize dopamine. And so I'm standing for TH, and here I'm standing for all the cells that my virus actually infected. And so what I'm able to do is then I can overlay those two images, and presuming that the things that are yellow have both the virus as well as all the machinery to make dopamine. And so now the assumption is that, okay, I'm getting a virus into about 90% of dopamine neurons in this region. And this is done with transgenic mice. So there are mice that you can breed that have certain genes that we can manipulate. But this is with rats that are non-transgenic. So this is a large finding, one that I was very surprised about. It was one of those things in grad school where you immediately cheer and then you take a week off. So, but all I've shown is that I can get it into cell bodies, but I haven't shown that I actually can get dopamine release. And so one thing that I've done now is I'm taking this purple line is actually indicating where I'm taking slices from. So now we take the rat brain, we keep the tissue alive, and we take little slivers of the brain tissue, and we can put it in a bath. And when we put it in the bath, we can use that fancy voltammetry technique to measure how much dopamine is released. And so when we shine blue light at it, which is this green trace here, we actually see that the dopamine is released as much as it is when we give an electrical stimulation. So now we're showing, one, that it is in tyrosine hydroxylase or dopamine neuron cell bodies, but we also can see it in these terminal boutons, and it is functional. We can get dopamine released when I shine blue light at it. Again, all because of green algae. And so now the idea is I can take these animals, I can implant these things over long periods of time, so I can inject the virus and plant this, and then I can train these animals to self-administer cocaine. And so now every time that the animal is going to go do a nose poke to receive cocaine, I can also shine blue light onto these neurons, onto these cell bodies. So now all I'm doing is, so that dip in dopamine, I'm now trying to increase it on a very fine resolution. And the way we're doing that is through our estimations, as we looked at the literature, and it seems like a response to dopamine is about 6 action potentials that we estimated that based on a lot of literature and my boss's many experiences in this field. And so what we're doing is we're just actually trying to cause about 6 action potentials by shining 6 pulses of light on these neurons, these neuron terminals every single time the animal nose pokes. And what I see is, I'm not showing all the controls here, but going into science, it is very important that you have controls. So I do have viruses that don't have all of these things, so I wanted to make sure that the virus didn't manipulate any of the drug-taking patterns. It does not. I have the data at the end of my talk if I have time, but this is the cool stuff and important things, I think, is that when I look at these non-escalated animals, and so what I'm showing here is a commutative plot of the amount of infusions that the animal had over an hour. So I'm giving the animal 1 hour of access to cocaine, and every single time the animal nose pokes, it just adds another 1 onto here. So every time the animal gets an infusion of cocaine, it adds 1 more. So at the end of that hour, the blue trace here is about 38. And so what I'm looking at here is the non-escalators and escalators. And you can clearly see that the escalators actually do have a decrease in their consumption when we stimulate every single time the animal nose pokes. Interestingly, the non-escalators actually had an increase in the amount of drug they're taking. Although that's not what we saw with L-DOPA, it is very interesting. There's multiple different reasons this is happening. We need to explore it further. I will point out that this also happens in other drugs of abuse with L-DOPA, which I might show at the end if I have time. But the idea here is that our hypothesis was that these phasic or these quick transient releases of dopamine, if you replace those, you actually can decrease the amount of cocaine the animal is taking. And so our thought is that this dopamine signal is, what I would call it as a feedback signal, is that it's letting the animal know, or it's letting the animal know that they don't need to take any more drug, or they become sated on the drug itself. And when that signal is going away, that feedback is lost, so the animal can possibly respond to get drug. But really, what I'm looking at now is that, one, phasic dopamine attenuates, so that dopamine response attenuates over the course of weeks of cocaine access. And we are able to use L-DOPA and/or optogenetics to reverse that escalated consumption. And so increasing dopamine decreases cocaine consumption. And so that checks off this spot, right? So now I'm saying, okay, so I know when these blue things go away, the animal's going to increase its consumption. All right, so the next thing I wanted to test was, well, what is the mechanisms that are actually altering dopamine signaling? And there's a lot of evidence. There's also some people that are on my committee, my dissertation committee, that were really pushing for me to study something called dynorphin, because they discovered the receptors that dynorphin binds to, so they're invested. Josh is the only one that's laughing because he knows who I'm talking about. Some others probably do as well. But the other reasons why I wanted to study dynorphin is that when animals are given injections, so this is just in their cavity, their stomach cavity, you can give them IP injections of cocaine. And if you do that over 10 days, you actually see that animals have an increase in the amount of dynorphin that is present. And so all this is measuring is prodynorphin mRNA, which is the precursor to dynorphin. And this says CORE, and I want to go back to this really quick. CORE just stands for cap opioid receptor, which is the receptor that dynorphin acts on. And so this is looking again at these dopamine traces, and when you give a mouse brain slice, when you wash on a kappa agonist onto this brain slice, you actually see a decrease in the amount of dopamine that's there. And then if you block that with a kappa antagonist, so you're basically you're making the receptor not able to bind to its agonist, you actually can bring dopamine release back up. So this is just showing that, okay, one, dynorphin's increasing when animals are getting cocaine chronically, and dynorphin is also able to bind to kappa opioid receptors and decrease dopamine release. And this right trace is just what happens when you actually knock kappa opioid receptors out of dopamine neurons. The agonist no longer has its effect. Okay. And so the idea here is, all right, well, one thing that we're looking at is dynorphin, my hypothesis is dynorphin on dopamine, but the best thing to study first is, well, how does dynorphin actually act on consumption? And so what if we could block dynorphin's actions by giving an antagonist directly into the nucleus accumbens? And so what I did is I implanted these animals with cannula, or little infusion tubes that go directly into the nucleus accumbens, and I can infuse a drug directly into that region. And so I infused a drug called Norvin-I, which is that antagonist, to block any of dynorphin's actions on the kappa opioid receptor. Norvin-I is an interesting drug because it lasts for a really long time. It actually lasts for 2 weeks because it takes that receptor and it makes it inactive, and then that receptor stays that way until it gets replaced until it basically gets replaced by receptor turnover, meaning that more receptors are shipped out to those terminals from the nucleus. Okay, so, and now what I'm doing is I'm just going to inject that drug right before these animals go into this extended access paradigm. So I'm training these animals, I implant this, the animals are trained so they know how to take cocaine, they learn the behavior, and we inject this drug. And what happens is that those animals that receive that drug, which is here in red, do not actually escalate their cocaine intake. So the idea here is, all right, we know that dynorphin and kappa opioid receptors are somehow mediating the consumption of cocaine. This again is just the statistics when you summarize all of their intake across weeks. And again, there's no significant difference in the Norvin-I animals, whereas the vehicle animals, vehicle just means a control infusion of no active drug, they actually do increase their amount of drug. Okay, so what I've shown is that, okay, so we know dynorphin and kappa do something to drug consumption, but I haven't really shown that it does anything to dopamine. And so my idea now was, well, what if I can attach those fancy electrodes that can record dopamine onto those infusion cannula, so I can now measure dopamine at the same exact place I am infusing drug into the brain. Okay, so the same concept, except now I'm just taking these recordings at the end of each one of those weeks, so I can record dopamine during this behavioral session. And what you find is that, what I found is that, again, animals that receive Norvin-I do not escalate their intake, whereas those animals that receive that control drug, the vehicle, did actually escalate their intake. Okay, so, and then when I look at these traces, I notice that you can see here across these weeks, so this is the baseline week, the post-injection week 1 and week 2, is that the animals that receive the vehicle drug actually have a decrease in their dopamine, which is mimicking or reinforcing the findings that I showed earlier, is that over time, over weeks, dopamine is decreasing at the same time as the animals are increasing their drug. Whereas the animals that receive Norvin-I do not have an increase. This is some preliminary data that I'm showing, so statistically it does not come out yet. I have other reasons for why that is, and I can talk a little bit about that towards the end, but I do want to just say one thing, is that I just spent 20 minutes convincing you that there's animals that escalate and animals that don't, and now I don't even care anymore, is that I'm just saying animals that receive the drug and animals that don't receive the drug. Meaning that both of my populations here are mixed. So my variance, or the spread of my data, is probably going to be very large, meaning I need a lot more animals to be able to statistically detect differences between them. And so one thing that we're doing, which is trying to predict which animals will be escalators before they actually receive the drug. This data is, or the technique that we're using to predict animals is machine learning algorithms, which is over my head, so I'm not going to talk too much about it. But currently using these machine learning algorithms, we actually have found that we can accurately define escalators with 79% accuracy. So there's still some off targets there that we need to fix, but it is something that we're interested in, and it is something that when you go to grad school is find people who do things. If you do go to grad school, find people who can do things that you can't, because they will help you. Because I don't know how to use machine learning algorithms, and it seems like it's working very well, and it's very promising. But basically what this data is showing now is that, just to summarize what I've shown you so far, is that the animals that we've inactivated the kappa through Norby and I prevents the development of escalation of cocaine intake. And then we also showing, so this current data, although it has not panned out yet statistically, is that the hypothesis that dynorphin is actually acting on kappa is actually mediating this decrease in dopamine. Our data does not disagree with that hypothesis. So I'm going to continue exploring this a little bit further, but using these machine learning algorithms so that now I can predict which animals are escalators and which animals are not. Okay, so now to aim 3, and aim 3 is a little bit less, so I basically have gone through these things of what I've been doing in my dissertation, and now I'm going to start talking about the things I'm currently doing and trying to problem solve through. And so my aim 3 is how does CRF, or corticotropin releasing factor, which is what CRF stands for, actually mediate the effects that we see in drug take. And what I mean by that is that we know that CRF is activating these medium spiny neurons, or those neurons that are located in the accumbens. And we also know that these neurons are releasing dynorphin. So we do know that CRF does act on these, and we do know that dynorphin is released from these neurons. We also know that when you actually give an animal CRF into the brain, it also increases the amount of kappa activation, meaning that you can detect kappa opioid receptors activation, and we know CRF also increases that. So it seems like everything's set up where CRF could be something that's actually activating and recruiting dynorphin into this schema I have laid out. And also, there's a paper in 2014 from George Kugel's lab that also had looked at when you give a CRF receptor 1 antagonist into the whole brain, you actually see that there's a decrease in the amount of cocaine that the animal intakes. These effects are mg per kg per session, so they look a little bit smaller than what our effects are, but they're quite similar. And so basically this is a dose-dependent idea, meaning that as you give more and more drug, there's actually more and more an effect, more and more of an effect of that drug on the amount of cocaine the animal's intaking. Okay, so now we know that CRF is somehow involved in this signal, but how it is involved is we're not entirely sure yet, and I'm still exploring that. But one thing is, rather than doing all of these drug manipulations, is CRF is also thought to be more mediated in things like incubation of craving and relapse. And so the idea is, well, when is CRF acting? I don't know. So although this data suggests that it's probably acting within the drug-taking session, is I also want to inhibit CRF acting on these receptors totally. And so one way to do this is, what if we can knock out these receptors out of the brain indefinitely? And so there's this new technique that's come out, I guess, probably in the last 3 years maybe, called CRISPR. And again, when science seems like it doesn't relate to you, and then it does, is that bacteria have this defense mechanism against viruses. So bacteria have DNA, and viruses can bind to bacteria, and what viruses do is they inject their DNA. And so if a bacteria, all of a sudden a virus binds onto a bacteria and injects its DNA, it could basically ruin that bacteria's life cycle. So bacteria have this really interesting defense mechanism called CRISPR. And what it does is it basically takes all those viral DNA that's injected, and it deletes them. It gets rid of them. And again, when science just amazes you, is that people have now been able to extract those mechanisms and use them, so now it's detecting genes that are in these animals, in these rats, and now it's deleting those genes out of the animal's DNA. And so we can package those mechanisms into a virus, and then I can deliver it into the brain of a rat. And so I did that, and so this is with the help of, I don't have their names up here, but Avery Humphrey and Larry Zweifel, who were at the University of Washington, basically came up with this way so I can inject this virus. It has the mechanisms where it detects the CRF receptor 1's DNA and reads it as a problem, as an invader. And so then it's going to delete those genes out of the DNA itself because it thinks it's a virus. And so now I can knock these receptors out. And what this picture is I took one rat, I injected the virus in one hemisphere of its brain, but not the other. And so all this red is doing right here is it's actually tagging that virus. So I've been able to fluoresce that virus, and I can show, well, in the hemisphere that I injected the virus, there's a lot of red, which makes sense. In the hemisphere where I did inject virus, it's completely black. I think there's some background there, but you can't see it. But you don't see any virus. That also makes sense. And so then what I did is I wanted, because this is just a functional test, can I do this, is then I injected CRF itself into that brain region. And so what you can do is you can, there's this thing called CFOS, and what CFOS does is it is a marker for neuronal activity. So if I inject this agonist into the brain, you would expect that there would be neuronal activity because it's acting on those receptors. And so then you can stain for CFOS. And so what I did is I injected I knocked out R1 out of one hemisphere and not the other, and then I injected CRF into the brain, and then I looked for CFOS staining. So what I'm looking at here is that I should see a decrease in CFOS staining due to CRF because I knocked its receptor out. And that's exactly what we see. So this is a very clever experiment that was thought of by a research scientist in Paul's lab, and the first time I did it, it worked out. This is one animal with 3 separate slices, so it does need to be done and replicated. But it does show that we are now able to actually manipulate this receptor in these animals. And so the plan moving forward, and what I'm actually working on right now, tomorrow I'm doing these surgeries, is to be able to knock this receptor out and then train these animals to self-administer cocaine. And so we can see how does this receptor actually regulate this. And so the idea here is that, one, I've shown you, this data has been parsed out and published, that we know that dopamine is effective consumption, so a decrease in dopamine is actually going to increase consumption. We have pretty convincing data that dynorphin is regulating dopamine, and that's also involved in this escalation of cocaine consumption. And now, what I'm currently working on is that CRF, which I think we have pretty cool tools. Again, I get to stand on the shoulders of giants and use these cool tools, and then I can explore how this CRF is affecting this signal. And so just to go, I just kind of talked all about this, but just to go through to make sure we're on the same page is that, one, dopamine's coming in, it's acting on these medium spiny neurons, and when you decrease dopamine, you're going to increase consumption. I believe that CRF is working through this medium spiny neuron, and then increasing dynorphin, which is mediating that effect. I apologize for not having the CRF on the slide. And so, at the beginning of the talk, I showed you a figure that talks about drug overdose deaths. The majority of those deaths, so I'm going to switch gears really quick, the majority of those deaths are not due to cocaine. They're due to opioids. And so one of the thoughts is that, all right, I thought about this, is this mechanism of dopamine actually, is it specific to psychostimulants and cocaine intake? So the idea is, well, what if we can do these same concepts and measure these same things, but in animals that are self-administering opioids? Yavin Shaham, which somebody knows very well, in the early 90s, in the early 90s, he used fentanyl and he put it into a liquid solution and saw that rats would drink it. And so the idea was, well, what if I could get rats to drink fentanyl, and I can study these same mechanisms? One, because that means I don't have to do a second surgery to implant an intravenous drug-eluting catheter, which could cause problems. And so one of the thoughts is, well, we can study this with a much more throughput, with a much lower attrition rate. And so the idea here is I've tried to mimic what we're doing. I'm sorry, this is pretty dark, but I'm trying to mimic as much as I possibly can how cocaine self-administration works, but with fentanyl. And so the idea is here there's a liquid delivery bowl, there's a light here that's working as a cue, there's an inactive port and an active port. And so when the animal nose pokes, which is right here where that dotted line, so when the DS, which means descriptive stimulus, when that ends, that means the animal nose poked, a nose poke light comes on, and another audio-visual stimulus also is presented. And during that time, there is a delivery of liquid fentanyl. So doing that, I actually had a lot of conversations with my boss about how to go about this and train and trick the animals into thinking that they should be drinking this liquid. And sometimes, as a graduate student, you just got to go with your gut. And I just put the animals in the box, and they started drinking it right off the bat. And very surprised. So what this is the inactive nose pokes, and this is the active nose pokes. So inactive's in orange, active's in black. And you can see that they don't really increase their intake, but they are only getting an 1 hour access a day. Previously, they've looked at, extended access to get animals to escalate on opioids is usually 12 hours rather than 6. So only giving these animals an hour of access, I didn't expect to see any escalation. But what happens is they are discriminating, which means that they are decreasing the amount of times that they're nose poking the inactive port compared to the amount of times that they're pressing or nose poking the active port, which is telling me, or at least hinting to me, that they're learning. We still need to think about exactly what the criteria is to say that these animals are learning, which is something that we're actively thinking about. One thing is this does start really high, is because when the animal nose pokes the inactive port, there's nothing happens, right? So imagine you have a light switch, and you just keep flicking it, and nothing's happening, and you have one that goes on and off every time you flick it. Well, you're probably going to test the one that doesn't do anything a billion times for no reason, because there's no consequence. So that's probably what's happening here, just to anthropomorphize a little bit. But yeah, so in my eyes, it looks like great. These animals are self-administering, or at least are drinking fentanyl. And so what I can look at is, I also looked at, I'm not showing it here, but I also can look at how much liquid is left over in the bowl at the end of the session, and I can detect exactly how much liquid the animal drank, because there's nowhere else for it to go. I guess they could be bathing in fentanyl liquid, but based on the videos that we have, it doesn't appear that way. And so now what I'm looking at, so rather than doing voltammetry, I wanted to start, well, is this behavior even L-DOPA sensitive? And so again, I'm looking at this commutative plot, and I see that animals that are, when they're given saline versus L-DOPA, the animals again decrease the amount of fentanyl they're taking. That's true for the active nose pokes and the mg per kg that they're consuming. So it appears that this behavior is also L-DOPA sensitive, and also means that dopamine is somehow involved in this behavior. Although we're not entirely sure if it's in the same brain region, because I haven't done anything in the brain. This is just systemic. We're injecting L-DOPA into the intraperitoneal cavity, and this is the effects that we see. One thing that's interesting, though, is when we look at how well L-DOPA reduced their taking versus what their baseline fentanyl intake was, we actually see that there's a positive correlation here, meaning that the more intake that the more fentanyl the animals are intaking, the better L-DOPA works, which is also similar to the way that what we saw with escalators and non-escalators in cocaine intake. What is also similar is that the animals that take very little baseline are actually increasing the amount of fentanyl they're intaking when we give them L-DOPA, which is something that we saw with optogenetics. So we do think that there is some kind of high-responding, low-responding effect of dopamine, and we're starting to look at what we really want to know is, what are the differences between those escalated and non-escalated animals? And that's why I'm studying all those neuropeptides. Because a lot of people are thinking, yeah, but what's the genetic differences between these high-responding escalated animals or high-responding, high-taking fentanyl animals? And the idea is it's most likely those peptides that I've been talking about. And so the future plan is also to do voltammetry in those animals that are self-administering fentanyl, so then we can start looking at what is the brain region, is it the nucleus accumbens, is it the certain specific location of the nucleus accumbens that we study, or is it somewhere else in the brain that dopamine's acting to decrease fentanyl consumption? So in conclusions, so what? That's kind of the idea, is that what we're really interested in is trying to figure out one way to think about this is these mechanisms that are controlling drug consumption are likely also setting up the aberrant signaling that's happening in the brain during abstinence. It could also be setting up an environment where relapse is likely to occur. But we also, one thing that we think about is everything I showed you is cocaine and fentanyl. And if I say I'm going to take a cocaine or fentanyl or opioid addict and turn them into a recreational user of cocaine and opioids, you're probably like, okay, that doesn't seem like a good idea. But we do have data that this also works with alcohol. And so right now at the University of Washington, we've done it with rats. I'm not showing that data, but L-DOPA also decreases alcohol intake. And so the idea is, what if you can turn an alcoholic into a social drinker? And so we've just moved that into clinical trials. There's a lab at the University of Washington called the Bar Lab, which is an actual bar that you walk into, and it's a one-way mirror, and you can like watch the people while they drink. I'm not directly involved with that, but it is based on the preclinical research that we've done with these things. So that's one application that it can go moving forward, but ultimately the idea would be if we can decrease the amount of drugs that a person takes, it will likely decrease the likelihood that they will overdose on drugs. Also, so just to quickly go through this, because it looks like I'm running out of time, is that moving forward, if you go in your science career, I've talked about these two things that are like, they seem like why would they matter to my research, and they did, and they're letting me study, so one, optogenetics, and two, CRISPR. So one thing, if I was starting off right now, and they are starting to make these things, is that neuropeptides are really, really hard to measure. So I showed you that I could measure dopamine, but neuropeptides are huge, and it's really hard to see the resolution or the signaling that the brain does with these quick release of neuropeptides. And so we have no idea what their temporal resolution is when they actually act in the brain. So there are these receptors that bind neuropeptides, and when they bind them, they fluoresce. The ones that I'm interested in, such as dynorphin and CRF, are not necessarily are fleshed out yet, or they're not working correctly, but it is something moving forward as you guys go forward in your science careers, start thinking about light, because it seems like fluorescence is where neuroscience is moving, and it's enabled us to detect all types of signals in the brain, as well as manipulate brain activity. And so with that, I will acknowledge my lab. So Paul Phillips is my PI. He's a great mentor. He allows me to come in the lab and not be in lab when I need to and don't need to be. I typically tend to be a 2 a.m. worker and not a noon worker, so he's a great mentor for that. Everybody highlighted here in blue are people that have helped me work on, have helped me or have led some of the projects that I talked about. And if I have 1 minute, if you're going to grad school, please have fun. That's my PSA. It's because a lot of people, it's really easy to get bogged down and lose sight of why you're actually here. I'm from Detroit, Michigan, and I moved to Denver and then moved here, and this is the most beautiful place I've been. And I've explored all over the Northwest. You live in a beautiful state. Please go explore it. And when you go to grad school, make sure you find an outlet. All right. Thank you.
We do have time for questions, if you guys have any. Yeah, Josh.
Awesome talk. Thank you. Just wondering how you reconcile your data and your conclusions with some of like the incentive sensitization models of addiction, where you often see increases in dopamine that drive drug seeking, and you're almost showing the opposite here. And just wondering how you think about those theories as well in conjunction with your data.
Yeah, so all the optogenetic stuff I showed is when the animal's actually taking drugs. And we have this whole side of these projects where we call it the diametric dopamine hypothesis, which is really just combining other people's hypotheses together in one context. And the way that we look at it is we actually see increases in dopamine when we show the animals the cue outside of a drug-taking session. So if you take that animal, you put it in the same box where it normally takes drug, and nose poking doesn't do anything, and they can't elicit those cues, but then all of a sudden you randomly show them that cue, dopamine spikes, and you can see that it also causes them to seek drug. And so what we're saying is that the same cue and the same it's the same cue but in different contexts can actually have opposite effects. So dopamine decrease is mediating the animal's response contingencies. So like as dopamine decreases, the animal's increasing their responding. But if you also just increase dopamine or stimulate dopamine, you can also cause them to start drug seeking. So if they're not in the context where they're already taking drugs and the cue is not initiated or elicited by their own behavior, it actually causes them to start seeking the drug. And so we've kind of thought about that as diametric dopamine because we've taken those same animals. So all the optogenetic stuff I've done, I've also just randomly stimulated dopamine when I randomly show them cues, and they start seeking drug. And so the idea is that it's both, but it has this fine temporal resolution and context-specific outcome on behavior. Yeah.
That was a really good explanation. I hadn't heard that diametric dopamine, putting that all together.
Yeah.
When you were talking about harm reduction, I think that's that concept of, there's so many things products, but it's going to be less. And it's a really important thing for people to kind of consider the ethics of that. And it's really important because people are looking into ways to do that. And so when you were looking at the L-DOPA, I was thinking about some of the proponents of using low-dose amphetamine for addicts. And I wonder, would you expect to see a similar effect with D-amphetamine, like really low dose versus your L-DOPA? I like the idea of just using L-DOPA.
Yeah.
But
I
there are
so
people out there pushing the amphetamine.
yeah, because there's like the replacement therapy is one of the thoughts of that. Yeah, I don't see why that wouldn't work. I guess one of the worries about that working is we thought L-DOPA was working because there was you're emptying the available pool of dopamine, but the optogenetics basically argues against that because we're still able to decrease. So I don't see why methamphetamine wouldn't work. I guess the addiction liability though of giving amphetamine versus L-DOPA to me would be the worrisome, just because.
That's where I think a lot of the pushback has come from. He's like, whoa.
Yeah, because L-DOPA is not. Yeah, L-DOPA, I would say the addiction liability of L-DOPA versus amphetamines, I would rather give somebody L-DOPA. But to connect the two is that one thing that I didn't explain is what Josh's question is also we need to make sure that L-DOPA doesn't increase relapse. And I think amphetamine probably would. The data, as far as in humans go, is what we can tell, L-DOPA really doesn't. So that is one aspect of why I would say I would prefer L-DOPA over methamphetamines or amphetamines, or just amphetamines. Yeah.
I realize you didn't do the work, but in determining the responders versus or the escalators versus the non-escalators,
Yeah.
it almost looked as though the non-escalators were starting to trend in the same direction. I wonder if anyone's taking that out a little further.
If you just take the non-escalators and give them more time to access drugs and see if they turn escalators. Not that I know of. There is a clear split in animals. I think Bruce Matt in the University of Colorado took animals out to like 70 days of self-administering cocaine with 1 hour access, and he also saw an equal split between animals that actually escalate their cocaine intake and don't. But he never continued further to see like what the population difference is. One thing that I will point out, because he mentioned it, is the reason that we wanted and first thought we could predict escalators and non-escalators based on training, is that non-escalators actually start higher in their cocaine intake and they just never change. And so the escalated animals actually just start lower, and then they increase their intake, and then they just continue to increase their intake. There's like no ceiling that we've been able to see. But the non-escalators don't really ever change from training or acquisition out. So it could be likely that if you give them more experience that they turn into escalators.
So you measured cocaine levels in blood?
No. We've used the model where we do brain cocaine concentrations, but usually around week 2 here, they're about equal, just as you would expect with most folks. But we've never done any blood work.
So the machine learning that you're doing, is this what you're using to predict What do you use?
Yeah, so I just deleted, and it used to be all right here what we were using, so I just deleted it. It was, so we're using their days, there's the number of sessions it takes to reach criteria, that they acquire the behavior, the active nose pokes that they do, the inactive nose pokes. I've looked at latency to first nose poke, and the inter-infusion intervals, which are going to be related to the active nose pokes. And I think those are the only aspects that we would put in it so far, but there's a lot of different behavioral ideas that we could put into that algorithm. One of the things that we're also wanting to do is that escalators start with more dopamine. Eventually we would start looking at predicting through neurochemical recordings rather than just behavior. We just don't have enough data to do that right now. Yeah.
Do these animals, do they binge their cocaine consumption, or do they front load it right when they get access? Like how, is there a temporal effect here that you're observing that specifically with the animals?
Yeah, so they actually load up and get there fast. So the fentanyl one actually shows it really well. Is that there's a point in which animals load up how much cocaine they take in the beginning of a session, and then they taper off. And so one way you can do is you can look at brain cocaine concentrations and use a learning function to basically say, when does that asymptote happen? And that's usually when you would say the animal's done with load up. So there's different stages in which animals take drugs. So as soon as they get in the box, they nose poke rapidly. You know, they have a 20-second period where nothing happens though because the cue's on. So they can't get any more drug during that time. I didn't explain that, sorry. But they can't get any more drug in that time. And they nose poke rapidly, and then once they get to a certain brain cocaine concentration, they just...
So at the beginning though, when they enter that box, do
Yeah.
you see a big spike in dopamine that then decreases as they consume?
So we have not looked at that, and that is something I am currently trying to code. There is a paper from Matt Wannett's lab that looked at the differences between load up and maintenance, and it seems like during load up the dopamine levels are lower. But they're not using extended access, they don't have escalators, and their behavioral assays are a little different. So I would still want to do it within ours, but yeah, that's a good question, we don't know. I mean, based on our hypothesis of low dopamine means excessive taking, I would expect, or I would hypothesize, because I can test it, is that I would hypothesize that there's low dopamine in the beginning of the session, and then they get to a higher amount of dopamine at the end of the session because they start responding less and less.
That's a fast scan you're saying?
Yeah.
No.